Clinical Virology Part 1. So significant advances in diagnostic methods have transformed the field of virology. Newer methods provide quicker turnaround times, test results are now more clinically useful, molecular diagnostic testing results in earlier intervention, earlier treatment, and better outcomes. So pretty much in the past we depended on cell culture and looking at how the cells were impacted if they were infected with patient samples. So we would take a sample from the patient, we would overlay it on a cell culture and see if it impacted the cell structure at all. And then we would kind of make the assumption that probably a virus was in that type of sample, especially if nothing bacterial or fungal ended up growing on the media. Then we developed the electron microscope, and we could see this electron microscope and we could see the organisms under the electron microscopes, and those were the viral particles that were growing inside the cells or around the cells. We also used to even inoculate animals or inoculate the certain parts of an egg. So the egg would get fertilized, and this egg would come from primarily chickens or turkeys. Turkey eggs were a little bit easier to work with because they're a little bit larger. But we would inject patient sample into a certain part of the egg, and then it would replicate and grow, and then we could use the electron microscope to see it. Well, those methods took a long time, and they were all kind of in the eye of the beholder. So if someone would look at cells and say, yeah, those don't look right, compared to another person's opinion, those were kind of subjective. So if we fast forward, now we've got molecular testing. And while we still say that cell culture is the gold standard for evaluating viruses and working with viruses, this molecular testing that we have now makes for a lot more rapid detection. And we're going to go through basic detection methods, and then we're going to start going through different classes of viruses in this lecture. So characteristics of viruses, this should be a review from a general microbiology class. So simple structure, nucleic acid is RNA or DNA. It's surrounded by a protein capsid, which is just the protein coat of that. Envelope, if it's an enveloped virus, it would have an envelope around it, and that envelope would consist of a phospholipid bilayer. And the purpose of the envelope is to allow for fusion when the virus infects our cells. Our cells are made up of a phospholipid bilayer. Viruses that are enveloped have that phospholipid bilayer inside or on their surface. So it allows for the fusion and incorporation of that virus into a cell. And if you recall, viruses make us sick because they get inside of our cells and replicate. They kind of shut down all of our cellular processes and force our cells to turn into new virus-producing factories. And different viruses have different routes of doing this, different modes and methods that they use to take over our cells, but that's basically what's going to happen. Nucleocapsid is the genome plus the protein capsid. So it's whatever the RNA or DNA is plus the protein capsid, and an entire virus particle is called a virion. There's morphological types of virions: helical, polyhedral, and complex. When present, the envelope masks the shape of the virion. Size extremes of human viruses. Pox viruses are the largest, poliovirus is the smallest. Viruses are classified in orders, families, genres, species, and based on genotype, RNA or DNA, and the presence or absence of an envelope. So if they have an envelope, they're called enveloped viruses, and if they don't, they're called naked viruses. Many viruses also have spike-like proteins that stick out, and those spike proteins are very specific for which types of cell surface receptors they can bind to. So typically, viruses can only infect certain types of cells. If you think about it, if you get the common cold, you typically can't give the common cold virus to your dog or cat at home. And that's because your dog and cat has different cell surface receptors that that virus can't really bind to. Now, viruses are evolving and they're ever changing, and they are developing the ability to jump across different species, hence the bird flu scare and a lot of these other types of viruses that are coming and becoming more prevalent. And we're seeing them cross the boundaries between species because they are generalizing the receptors that they can bind to. Nucleotide sequence is also a valuable tool for the taxonomic placement of viruses. So this is a list of viruses causing human diseases. We're going to go through some specifically throughout this lecture series. Viruses, as I mentioned, are obligate intracellular parasites, which means they have to get inside of your cells in order to replicate. Steps of infection, you have step 1, absorption or attachment to the cell surface. There's specific cell receptors that viral adhesion molecules are going to be able to bind to. Most host cell receptors are glycoproteins. The adhesion molecule receptor interaction determines the viral tissue tropism in the host. Then we have penetration. So naked viruses direct penetration through the cell membrane. Enveloped viruses are going to fuse with the cell membrane, and that's because of that phospholipid bilayer that I talked about. And then endocytosis, they enter the cell in a cytoplasmic vacuole. Along with penetration comes uncoating. So uncoating is when that virus loses its protein coat and releases the genome. RNA viruses release their genome into the cytoplasm. DNA viruses release their genome into the host nucleus. Replication and translation. The viral genome then directs the host cell to make viral proteins and replicate the viral genome. Metabolism of the host cell may be completely stopped or continue at a reduced scale. Then step 3, assembly or maturation of the virus particles. So the capsid protein subunits aggregate to form capsimers, and then capsimers combine to form the capsid. The capsid and genome associate to form the nucleocapsid. And then you have release. Naked viruses are released by cell lysis. In other words, the cell is just going to rupture and all the viral particles are going to be released. Enveloped viruses are released by budding. During budding, part of the host cell membrane surrounds the viral capsid and becomes the viral envelope. This takes a chunk of the host cell every time that it buds off, and if enough viral particles were made inside of the cell, there's going to be nothing left of the cell, so eventually the cell shrivels down to nothing. Now, it's this process that causes a lot of our symptoms when we have viral infections. It's the slow deterioration of our cells or the rapid lysis of our cells that stimulates the inflammatory response. So typically in a viral infection, we're going to have massive cell death in the area that the infection is taking place, and that cell death is going to elicit the inflammatory response. Laboratory diagnosis of viral infections, varying levels of viral testing are available. So there's rapid tests where we use antibodies against particular viruses, and we can detect them that way. Detection of RNA or DNA using PCR assays, so molecular testing. And then cell cultures are available only in full-service virology labs. So you typically wouldn't see cell culture happening in your run-of-the-mill clinical lab. Clinical signs and symptoms of the disease often point to the target organ that's involved, which can help determine the most appropriate specimens to collect. This, combined with basic understanding of viral pathogenesis, can help in specimen selection. It's important to ensure, however, the specimen collected can be used to isolate a wide range of viral pathogens because different viruses have overlapping clinical symptoms. The best specimens are collected as early as possible, which in many infections is even before symptoms occur. The sensitivity of viral culture can decrease rapidly 3 days after the acute onset of symptoms. Care must be taken to collect specimens appropriately to maximize detection and identification. Specimens should be collected aseptically, just like if you're going to collect a specimen for bacterial culture. Depending on anatomic sites, the method of collection can be sterile or non-sterile. Example of a non-sterile, contamination with bacteria and or fungi. Non-culture-based test methods are typically not affected by contamination, but this varies with the system. In a normally sterile specimen, such as blood, CSF, or tissue, identification of a virus without bacterial isolates usually means that the virus is actually causing the disease. Non-sterile specimens are obtained from sites that contain normal biota, such as the respiratory tract, genital tract, skin, or stool. These specimens may require processing to reduce contaminants and promote viral growth. Aspiration or aspirated secretions are often preferable, but swabs are easier to use for collection. Swabs may be made of dicron or rayon. Calcium alginate swabs inhibit the replication of some viruses and can interfere with NATs, or nucleic acid amplification tests. Flocked swab is recommended. Tissue samples must be kept moist and must not be placed in transport media unless it's specifically designed for viral preservation. Substances that can be added to sterile containers to keep tissues from drying out include viral transport medium, saline, trypticase soy broth. Antibacterial and antifungal agents can also be added to these samples. Samples should be collected without viral transport media include blood, bone marrow, CSF, amniotic fluid, urine, pericardial fluid, and pleural fluid. Refrigerate if necessary at 4 degrees Celsius. Freeze only if it's longer than 4 days. Never freeze specimens at -20 because this temperature facilitates the formation of ice crystals that will disrupt the host cells and result in loss of viral viability. Repeated freeze-thaw cycles can also be an issue. Examples of specimen types are listed here. And then there's a table that can show tests available for common viral pathogens and specimens for culture. So diagnostic categories: direct viral detection in clinical specimens, isolation of viruses in cell cultures, serological assays to detect antibodies to the virus, so that can tell us if you've been exposed to a particular virus, major methods for direct viral detection in clinical specimens, microscopy to detect cytopathic effect, detection of viral antigens, and nucleic acid detection. Not usually as sensitive as culture methods, can offer quick results to allow rapid therapy. Many tests can be performed in a matter of a few minutes. Viral detection allows clinicians to make relevant decisions about therapy, infection control measures, and hospitalization. In many cases, virology results may be available before routine bacteriology culture results. Bright field microscopy is not useful in looking at viruses because viruses are super tiny, so typically we have to look at electron microscopy. It can be used to visualize virions and is useful to detect non-culturable viruses such as the Norwalk virus in stool filtrates. It's expensive, labor-intensive, and not a very sensitive method of detecting viruses. Therefore, it's rarely used in clinical labs, more suited for large teaching or research institutions. Cytopathic effect, though, when viruses impact or get inside of your cells, sometimes they cause the cells to take different shapes. They can cause different types of what they call inclusion bodies to form. So inclusion bodies are just like garbage that accumulates inside of your cells, and that garbage is going to be a bunch of different viral components. Distinctive visual changes in infected cells, CPE, can be viewed using bright field microscopy. A sand smear is a common method used, scraping of an ulcer base to look for cells, large round keratinocytes that are degenerated as a result of viral infection. Can detect Cowdrey type A bodies from herpes simplex virus and varicella zoster virus lesions. Pap smears can reveal human papillomavirus associated chyliocytes, squamous epithelial cells with an enlarged nucleus surrounded by a non-staining halo. Direct fluorescent antibody tests are available in EIAs. And here's a picture of some of those immunochromatographic tests. Nucleic acid-based detection shifted the focus of clinical virology. Not only can the presence or absence of a virus be determined with nucleic acid-based analysis, but depending on the assay used, a quantitative test or result can also be obtained, so we can see viral load or how many viral particles are there. There are many advantages, including much faster turnaround time. Disadvantages: detection of both active and inactive viruses, higher cost, need for specialized training and more complex facilities, lack of assays approved by the FDA, and smaller clinical laboratories often rely on sending these tests to reference laboratories at a higher cost. Examples are listed here. Isolating viruses is still the gold standard against which all other methods are compared. 3 methods are used for viral isolation: cell culture, which is the most commonly used; animal inoculation, we inject it into an animal; and embryonid eggs. So that's when an egg gets fertilized, typically a chicken or a turkey egg, and then we inject the virus into the rapidly dividing portion of the egg. Unfortunately, that does kill the developing baby inside of the egg, but that's one method that we use to evaluate viruses or grow them. Cell culture is used to indicate culture of cells in vitro, so in a petri dish. Tissue culture or organ culture is used to denote the growth of tissues or an organ so the architecture or function of the tissue or organ is preserved. Cell viability is maintained periodically by removing cells from the surface, diluting them, and placing them into a new container. When you start cell culture, it is a time consuming process. It's like taking care of a newborn baby. So these cells have to stay alive and they're constantly dividing. Now, as they divide, they're going to produce metabolites, and the metabolites that come off of cells are usually acidic. So, a lot of times when you culture cells, you are going to be culturing them in media that has a pH indicator. And as soon as the media with the pH indicator turns a certain color, then you know it's time to wash the cells and put fresh media in there. So what we do during that process is typically the solution we put them in initially is going to be pink. And then as the pH drops, as it gets more acidic, it's going to turn more yellow in color. So once it gets to be that like yellow crummy color, we are going to treat the cells with a chemical that get them to stick to the plastic petri dish. Then we dump out the solution. Then we wash the cells, and then we resuspend them in that pink solution. And during that process, we have to be very delicate with the cells and very gentle because these are typically eukaryotic cells that we're going to be growing, eukaryotic like animal cells, human cells, and they are very fragile. So they don't have like a tough cell wall like you'd see with bacteria or with fungi. So they're very easy to rupture. So we have to be very careful with how we treat them with the chemicals. This process is referred to as splitting or passaging. Splitting cells is another thing that we can do. So we can just wash and replace the media, but we also, during that process, we count how many cells are in a particular area, and then we can calculate based on what we saw in that area, and we can apply it to the whole culture. If our cell numbers get too high, then we actually have to split the cells. So we would take half of the cells and put them into a fresh petri dish and then allow them to grow. Now, if our goal is not to just continuously propagate more and more cells, we can actually discard half of the cells and just leave the other half to grow. But either way, eventually the cells are going to divide and they're going to crowd each other out. So we have to take the cells and split them into two separate petri dishes. Some cell lines can only be passaged a few times before new cells must be obtained. Some viruses produce characteristic CPE that can provide a presumptive identification of a virus isolated from a clinical specimen. Primary cell cultures, the tissue is finely minced and then treated with an enzyme such as trypsin to disperse individual cells. The cells are then seeded onto a surface, such as in a flask or test tube, to form a monolayer. With primary cell lines, only minimal cell division occurs. Primary cell monolayers can only be passaged a few times. Finite cell cultures can divide, but passage is limited to about 50 generations. Finite cell lines, like primary cell lines, are diploid. That is, they contain 2 copies of each chromosome. Diploid is the genetic makeup for eukaryotic cells. As the number of passages increases, the cells become more insensitive to viral infection. Continuous cell cultures, capable of infinite passage and are heteroploid. They have abnormal and variable numbers of chromosomes. Sometimes the number is not a multiple of the normal haploid number. It's referred to a nucleoid. HEP2 derived from human laryngeal epidermoid carcinoma. A549 derived from human lung carcinoma. And Viro derived from monkey kidneys. HeLa is another one that I added there, Henrietta Lack cells. So Henrietta Lack was a woman that developed, I believe it was cervical cancer. Cells were isolated from her cervix about 100 some plus years ago, and they were frozen. Now these cells have divided continuously for 100 plus years. They're cancer cells, and they don't stop dividing. So what we did is we just let them divide, and we fractioned them off. And now labs all over the world use the cells from this one woman's cervix, one woman's cervical cancer, that years and years later, they're still dividing and still allowing us to do cell culture. The benefit of these is that they don't stop dividing, so you can keep using them and keep looking at them. The downfall is sometimes they aren't as easily infected with viruses, and sometimes they have some limitations because they are cancer cells. So, pros and cons of using each type of those cells, but if we have a continuous cell culture, we're primarily looking at cancer cells that just don't stop dividing. Here's some cell cultures commonly used in the clinical virology lab. Mixed or engineered cell cultures are cell lines that contain a mixture of two different cell types. They're made up of cells genetically modified to make identification of viral infection easier. Mixed cell lines have been developed by combining two cell lines susceptible to certain types of viruses, such as respiratory or enteric viruses. Greater sensitivity to a wider range of viruses, and useful for fastidious viruses. Screening cell cultures for viruses. Some viruses produce that characteristic cell or cytopathic effect. Fluorescent antibodies can be used, and different types of immunological or molecular tests can be used. Centrifugation enhanced shell vial culture. Cells are grown on a round cover slip in a shell vial covered in culture media. Shell vial is a small, round, flat-bottomed tube, generally with a screw cap. Sample is added to the shell vial, and centrifugation is used to promote viral absorption to the cells. The cover slip is removed, and an immunofluorescent assay is performed on the slide. Based on the type of clinical specimen and suspected virus, a variety of fluorescent-labeled antibodies can be used. Modification of this procedure can use a flat-bottomed microtiter wells. Although this is better than looking for cytopathic effects, in many cases it can be labor-intensive, and often cultures are done in duplicate, which results in reading at 24 hours and then again at 48 hours, thus increasing the TAT. Viral serology detects circulating antibodies to viruses after exposure. This is a problem because if a vaccine exists against that virus, then you detect the antibody. It could just mean that you're detecting the antibodies against the vaccine. Also, it doesn't indicate a current infection. It could indicate a previous infection. Second, antibody producing capabilities of human hosts differ widely from person to person. With few exceptions, paired sera, acute and convalescent, demonstrating subconversion or fourfold rise in titer are required to establish a diagnosis of a recent infection. Serological assays are usually retrospective. Some assays can distinguish IgM from IgG. IgM indicates a recent infection. IgG means you've got immunity against it for a future infection. Indications for serological testing: diagnosis of infections with non-culturable agents such as hepatitis viruses, distinguishing past IgG or acute current IgM viral infection, determination of immune status regarding rubella virus, measles, varicella, hepatitis A and hepatitis B. Monitoring patients who are immunocompromised, epidemiologic or prevalent studies. Disadvantages: sometimes there's a lack of paired sera. Antibodies are not always diagnostic. Does not work well in patients who are severely immunocompromised. Reactivating viruses have previously induced antibody formation. Infants with maternal antibody, cross-reactivity with other antibodies. So now we're going to move on to some specific viruses. So viruses, as I mentioned previously, can be categorized based on their nucleic acid. Excuse me. So we're going to talk first about double-stranded DNA viruses, or dsDNA viruses. And we're only going to focus on the very clinically relevant ones. So members are adenoviridae, the adenovirus, herpes viridae, HSV 1 and 2, CMV, EBV, VZV, human herpes 6, 7, and 8. And then papillomaviridae and poxviridae are also part of this category. Adenoviridae, naked icosahedral viruses with double-stranded DNA. Now there's going to be a lot of information presented in these slides. The main things to focus on are the diseases it causes, any common names associated with the virus, and the diagnostics. You can also, if diseases are caused, you can kind of make note of the symptoms of the diseases. So common serotypes you won't need to have memorized. Clinical presentations, about 50% are asymptomatic, 10% pneumonia, 5 to 15% gastroenteritis in children particularly, and they're responsible for epidemic keratoconjunctivitis, acute hemorrhagic cystitis, and pharyngeoconjunctival fever. Isolation methods, they can be identified but not serotyped by EIA. Commercial antigen detection kits are available. Fluorescent antibody methods, nucleic acid tests, and serotyping. And here's a picture. You will not need to be able to identify pictures of these viruses. Herpes viridase, so the genome of linear dsDNA, icosahedral capsid, amorphous integument surrounding the capsid, and envelope. There's 8 species of human herpes viruses, HSV1 and 2, varicella zoster virus, Epstein-Barr virus, human cytomegalovirus, HHV6, 7, 8, and HHV8 is Kaposi's sarcoma herpes virus. So talking about the first two, herpes simplex virus, HSV1 and HSV2. HSV1 is also called human herpes virus 1, HSV2, human herpes virus 2. You do need to know what HHV and HSV stand for. These abbreviations for these viruses is going to be important for the qualifying exam. HSV infections are very common by adulthood. Approximately 80% of Americans have been affected with HSV1 and 20% have had HSV2 infections. These figures indicate that about 1 in 6 persons in the United States has had an HSV infection, and most infections are asymptomatic. Types of infections. HSV infections occur worldwide, and infections cause a wide spectrum of clinical manifestations. HSV 1 and 2 cause the same diseases. So previously, we used to think HSV 1 would just cause oral herpes, and HSV 2 would just cause genital herpes. But now we know that each can cause both. Skin vesicles are filled with clear fluid and have a red base. The vesicles progress to pustular lesions, ulcerate, and then finally crust over. So they burst open, some clear fluid can emerge, and then they're going to crust over and heal. They can be very painful. Oral herpes. Most oral herpes infections are caused by HSV1, but many are also caused by HSV2, as I mentioned. Incubation, 2 days to 2 weeks from the time you actually get infected to the time you show symptoms. Primary infections are usually asymptomatic, so the first time you get infected, you usually don't have symptoms. Recurrent reactivation of HSV borders the lips. This infection can keep coming back throughout the course of the lifetime. An early symptom of burning or pain followed by the vesicle ulcer and crusted lesions is the typical pattern. These eruptions can result from stress or sunlight and are often quite painful. Someone that goes in for oral surgery, or I've heard of people that have like teeth removed and then all of a sudden they get a massive outbreak of oral herpes in their mouth. So that's stress in the area that can cause a huge outbreak. Genital herpes. Infections are usually caused by HSV2, although as I mentioned, HSV1 can also cause as many as 1/3 of the infections. Many individuals have antibodies to HSV2 and have not been diagnosed with genital herpes. In women, vesicles on both sides of the labia or involvement of the cervix and vulva is possible. Genital herpes, affected sites, penis shaft, glands, and prepuce of the penis. In women and men, urethral involvement. Recurrent herpes infections involve the same sites as primary infections. However, the urethra is less commonly involved with fewer symptoms and shorter duration. Herpes genitals can much as double the risk of sexual transmission of HIV. Neonatal herpes is more severe than HSV, or HSV2 is more severe than HSV1 in neonates, acquired in utero during birth or after birth. Most commonly during a vaginal delivery, there's a 50% chance of transmission when the mother has a primary infection. So if the mother has a primary infection, so you're seeing open lesions on the vagina or the labia, it's highly recommended that the baby is not delivered by vaginal delivery. The risk of transmission is very low when the mother has recurrent herpes. Cesarean delivery or suppressive antiviral therapy at delivery significantly reduces the transmission. Mortality associated with disseminated and neonatal disease is about 60% in treated neonates and exceeds 70% in untreated neonates. HSV encephalitis, devastating disease with a mortality rate of 70%, accounts for 20% of all cases in the United States. Encephalitis is usually caused by HSV2 in neonates and HSV1 in older children and adults. It's also associated with an immunocompromised status. Survival rates and clinical outcomes greatly improved with IV antiviral treatment. Ocular herpes, infection of the conjunctiva, swelling of the eyelids associated with fluid-filled vesicles. Corneal involvement, herpes keratitis, can result from destructive ulceration and perforation of the cornea, leading to blindness. Fortunately, most infections involve only the superficial epithelial layer and heal completely with treatment. Diagnosis is made by antigen detection or viral isolation in culture. Smaller laboratories will look for cytopathic effects and, if present, specific fluorescent antibodies to HSV1 or 2. Other specimen types stained and observed for cytopathic effects consistent with HIV infection. And then here's just some pictures. Culture is still considered the gold standard. The best specimens for culture are aspirates from the vesicle, open lesions, or host cells. Now this means that we have to get a needle inside of a vesicle and draw the fluid out, which can be extremely painful. Sometimes they take a swab and try to burst open the lesion to get the fluid out. Again, extremely painful. To make a culture-confirmed diagnosis of encephalitis, brain biopsy material is required. The preferred method is in CSF is PCR. In many studies, gene amplification for HSV and CSF approaches 100% sensitivity. There's other newer nucleic acid tests. The diagnostic panel is available for the detection of HSV1, 2, and several other viruses and bacteria that cause meningitis. Culture. HSV replicates rapidly. CPE can be seen within 24 hours. Tests that differentiate antibody responses to HSV are listed below. And then we're moving on to cytomegalovirus. So cytomegalovirus, CMV, again, another abbreviation that you're going to need to know the what it stands for, also known as human herpes virus 5. You need to be able to associate the number with what virus it is. The name originates from the enlargement of infected cells. Cyto is Latin for cell and mega means large. Spread by close contact with an infected person via tears, saliva, urine, stool, breast milk, semen, vaginal, and cervical secretions, blood, and blood products. CMV infection is the most common congenital infection in the United States. Most infections are asymptomatic in the immunocompetent host. Can be a self-limiting mononucleosis-like illness with fever and hepatitis. In immunocompromised hosts, such as transplant recipients and patients with HIV, can become a significant life-threatening systematic disease involving almost any organ. Congenital infections are often asymptomatic but can present with serious manifestation if the mom acquires primary infection during pregnancy. It's unlikely to occur if the mom was seropositive at the time of conception. Symptoms are listed below. This is one of the leading causes of deafness and intellectual impairment in the neonate. Detection, best confirmed by the isolation of the virus from normally sterile body fluids, can be cultured from urine or respiratory specimens. CMV is a typical herpes virus, but it replicates only in human cells and more slowly compared with HSV or VZV. Can be isolated in cell culture only by using human diploid fibroblast cell. May take up to 3 weeks for cytopathic effects to appear, and serology is not helpful. So here are some images. Viral antigenemia tests, specific, sensitive, rapid, and relatively easy to perform. However, several newer NATs may replace this test. Molecular-based testing is widely used to detect virus particles in clinical samples. Multiplex PCR is becoming a lot more common, where one PCR reaction can detect multiple types of viruses. And NATs are the preferred method for determining viral load. Moving on to Epstein-Barr virus, EBV. EBV is also known as HHV4. It causes infectious mononucleosis. Up to 95% of adults between ages 35 and 40 have been infected. Children can become infected with few signs of infection. Signs include sore throat, fever, lymphadenopathy, hepatomegaly, splenomegaly, and general malaise. It's usually the tiredness that can last the most. The most severe symptom is the overall fatigue. Symptoms usually resolve in a few weeks, except for the tiredness, the fatigue that can last for much, much longer. They typically say like a month. Once you get infected with mono, you can be extremely exhausted for about a month. Complications of EBV include splenic hemorrhage and rupture, hepatitis, thrombocytopenia, purpura with hemolytic anemia, Reye syndrome, encephalitis, or other neurological syndromes. Some cancers have been associated with EBV: Burkitt lymphoma, Hodgkin's disease, and nasopharyngeal carcinoma. EBV has been increasingly recognized as an important infectious agent in transplant patients. Incubation period ranges from 2 days to 2 weeks. Most adults demonstrate EBV antibodies. Children are almost always symptomatic. As the age at the time of infection increases to young adulthood, a corresponding increase occurs in the ratio of symptomatic to asymptomatic infections. Diagnosis: viral culture requires human B lymphocytes, and that's pretty difficult to culture. So it's usually beyond the capabilities of most clinical virology labs. Laboratory diagnosis of EBV is often accomplished with serological tests. Rapid test kits for detecting heterophile antibodies exist. And then there's EBV-specific serological tests. And here's the interpretation. And serological evaluation of Epstein-Barr virus infection is depicted here. And moving on to varicella-zoster virus. Varicella-zoster virus, VZV, also known as HHV3, spreads by droplet inhalation or direct contact with infectious lesions. Cell-free virus is produced at very high levels in the skin vesicles. Vesicle fluid is highly infectious. There's 2 clinical manifestations: varicella, which is chickenpox, or zoster, which is shingles. In the United States, more than 90% of adults have antibodies to VZV. Varicella is the primary infection and is highly contagious. Infections generally clinically apparent. Varicella symptoms: mild febrile illness, rash, vesicular lesions. Lesion progresses to fluid in lesions, it dries, lesions crust over, and heal in 1 to 2 weeks. Painful oral mucosa lesions may develop, particularly in adults. Diagnosis is based on clinical findings. Smaller labs may use smears to look for cytopathic effects. VZV can be cultured on human embryonic lung or vero cells. NATs such as PCR have become the standard for diagnosis. Vaccines are available for chickenpox and zoster. Antiviral treatment is available. And then moving on to HHV6. It's in the genus Roseolavirus. 2 variants, A and B. They're indistinguishable serologically. Variant B seems to be the cause of disease. Common pathogen: about 95% of young adults are seropositive. The virus persists in the salivary glands and has been associated with stool specimens, but evidence indicates that saliva is the most likely route of transmission. Inhalation of respiratory droplets and close contact with infected individuals is the primary portal of entry. Associated with the childhood disease roseola, also known as roseola infantum, exanthum subitum, sixth disease. Sixth disease is the one that I've most commonly heard it referred to as. Children are protected by maternal antibodies until approximately 6 months of age. And then there's the seroconversion occurs in 90% of children between 6 months and 2 years of age. In immunocompetent individuals, most infections are mild or asymptomatic. When symptoms occur, the disease is acute and febrile. A macropapular rash appears as the fever resolves. 30 to 40% of infected children with symptoms experience seizures. Sometimes These are known as febrile seizures. So the fever happens, it increases the body temperature, and then causes the seizure. There may be a connection to the development of progressive multifocal leukoencephalopathy and multiple sclerosis. Diagnosis is usually made clinically. Isolation of the virus can happen. Serology, if paired sera are available. PCR and viral load testing offer the most sensitive and specific means for diagnosing primary infection. HHV7 is the gene in the genus Roseolavirus, just like HHV6. The CD4 molecule serves as the receptor for it, and it infects T lymphocytes. It also has other receptors and a broad range of host cells. Extremely common, it's shed in the saliva of about 75% of adults, causes roseola, clinically identical to HHV6. Causes latent infections in T lymphocytes. Most 2-year-olds are seronegative for HHV7. Specimens for isolation in culture would be peripheral blood lymphocytes or cord blood lymphocytes, can be isolated from the saliva of healthy individuals, rarely isolated from peripheral blood and mononuclear cells. PCR assays can detect the virus, but interpreting results can be problematic. Serological tests are susceptible to cross reactions. And then HHV8. In the genus Radenovirus, can be detected in all forms of Kaposi sarcoma. Kaposi sarcoma is a type of cancer that is prevalent in HIV patients. So the common name for the virus is Kaposi sarcoma-associated herpes virus. This has been shown to play a role in the development of primary effusion lymphomas and multicentric Castleman disease. In North America and in much of Europe, appears to be transmitted primarily through sexual contact. In Africa, transmission is by more casual means is more likely. Pattern of infection is like that of HSV2, although men who have sex with men seem to be more susceptible than heterosexuals. Diagnosis, it can't be recovered in culture, so PCR has been shown to be less sensitive than some immunoassays. In situ hybridization can detect HHV-affected tissue. Commercial monoclonal antibodies, identification of HHV8 infected cells in various types of lesions by immunohistochemistry is more common. Serological tests being evaluated and may soon be available.